Hello and welcome back, everyone. My name is Delaram. I just defended my PhD a few months ago.
Congrats.
Thank you. I studied computational biology, and I was in Gary Bader's lab working on statistical and like ML methods for single-cell transcriptomics data. I've taught this module, I think 2 years ago, right? And so this is the second time I'm covering this. So today we're going to discuss dimensionality reduction methods, what they are, why do we bother with them, and what are some of the popular methods for performing this task. All right, so first of all, let's talk about some of the terminology that we're using here. What do we mean by dimension? So in today's discussion, whenever I talk about like dimension, I'm basically referring to a variable. So for example, in any data set that you were looking at, so what was the variables in the mouse data sets that Shraddha was just covering for you guys? What was the variable in that data set? So in any data set that you're dealing with, you have variable and you have samples, right? We usually deal with matrix format data sets. And what was the variable? Variable is essentially what we're measuring, right? So what was our variable in the data set that so expression is the exact measurement. So it's essentially if you think you have a matrix. I like Blackboard, by the way. I'm gonna be here frequently. Okay, so we have a matrix. Each of these elements are a measurement, right? So what was this measurement?
Microarray.
Yeah, microarray, but essentially represent gene expression, right? Yeah, exactly. It could be like we use different proxies for measuring genes, right? But usually this matrix has some columns and it has some rows.
Yeah.
So as in columns, in best practices, we usually represent variables or features. And in rows, we usually represent samples.
Is that why we have to transpose things?
Yes, yeah, we're actually gonna see that again. So there are different
Because the data sets are the other way, right? Like the data sets are like
It
samples on the
depends.
top.
It depends. Like they come in different versions.
Okay.
There are also different kind of best practices in R versus Python. So it depends on what you're working with. But I would say this is the more traditional way of representing the data historically. We often have the measurement, like the variables or features as columns and rows represent samples. So in microarray or I don't know, bulk RNA-seq, what will be our variables and what would be our samples? And what would be each of these elements? So in a microarray or bulk RNA-seq, could you tell me what the variable or features are? Genes, right? Not gene expressions, because expression is essentially how much of this variable we have in this cell.
Values.
Right, exactly. Those are the values. So we have genes as variables, and as samples, we have essentially, I don't know, biopsies or whatever.
Yeah, they don't tell us, it's just M1.
Yeah, exactly. Patient samples, they could be whatever. So do you know what these two will be in a single cell RNA-seq data? So genes is the same, right? For in a lot, essentially in anything that we're measuring gene expression, the variable or the features will be genes.
This single cell is single cells.
Exactly. So instead of like a tissue from a patient, we have individual cells. Right. Okay. So what do you mean when I'm saying dimension? What I'm essentially referring to is this part, the variables or features. So what would be the dimension of this matrix when we're kind of like thinking of it more statistically? Of course, it's a 2 by, like it's a sample by feature matrix, right? But then usually the terminology that today we're gonna discuss here, when I talk about the dimensionality of this data set, I'm referring to the features, the number of features. So what would be in a microarray data set? What would be the dimensionality of that?
Number of genes.
Number of genes, right? So this is essentially a major issue in genomics or any sort of omics data set that you deal with, because it's kind of like the same if you're dealing with proteins or SNPs or whatever. Usually the data sets that we're dealing with have a large number of variables or features, right? How many genes approximately do we have in human genome? Like 2,000 to 30,000, right? Or like we have millions of SNPs, if you're handling some, I don't know, SNP data set, like UK Biobank, whatever. So this is generally a major issue in computational biology, that you have too many of these variables, and you want to, for scalability and noise issues, you would like to have a smaller data set. Like represent this data in a way that the number of features that you're dealing with will be smaller. And that's essentially the topic of today's discussion. Like that's it. We want to go from high dimensions to low dimensions, from high number of features to lower number of features, right? Okay, so this is just a simple example again. So we were looking at some gene expression in mouse data sets. So again, what are the features and samples here? Features are genes. Yep. Yeah, but this is transpose of that, right? So don't just directly think about rows versus columns. Think about what is the point of this study and what are we measuring, right? So the variable here is again genes. So, okay, so what would be the dimension of features here? 2, exactly. So we have 2 genes, right? And we have 6 mice. So 6 samples. Genes are variables, mice are samples. And again, this is not restricted to microarray data. We can have like single cell proteomics data set. Okay, here, what are our variables? Yeah, proteins. And we're measuring them in individual cells. So cells are our samples. And this issue is like dimensional rejection. It's a very broad problem. It's not restricted to biology in any ways. So you can have, I don't know, from like a data set of Netflix users where you have your samples would be different account, yeah, exactly, different users, and your features could be like the movies that they liked, and the values that you're measuring is like the score that they gave to that specific movie, right? Here we have another one for students and topics that they're studying, and you know the scores could represent, for example, their exam scores or whatever. All right, so we want to talk about, okay, how can we visualize differences in samples? So in this data set, in the first one, when we have only one variable, it's pretty straightforward, right? So we essentially have 6 different numbers. We can define a coordinate system. We have one line, and we put dots for measurements, right? So in the middle, you can think that it's, I don't know, not in the middle here, but you start from 0 to whatever number and just put dots for that represent expression of a gene for that specific mice. So what if we want to plot this on 2 dimensions? Then we have gene 1 and gene 2, right? And each of the dots, again, what is each dot here in this example? Example. What's the sample? Right. So again, dots here in this specific example, they're not representing genes, right? They're representing mice. That's the important part. And what do you notice here in the pattern of the, yeah, there are two clusters, right? So visualization is generally helpful for tasks like that. You can do a quick evaluation, exploratory data analysis, and figure out what are the patterns that are being represented by my data set. Okay, so x-axis is gene 1, y-axis is gene 2. And we can go on and add axes here, right? So we can represent gene number 3 if we were measuring it and kind of like plot it in a 3-dimensional plot. But sadly, human brain cannot really comprehend plots higher than 3 dimensions. 3 is even sometimes a little bit tricky for us. And in many computational biology problems, we're dealing with thousands or millions of features. So the problem is, okay, how can we represent the data when we're dealing with so many number of variables? And that's when dimensionality reduction becomes useful. So essentially the idea is that let's transform the data to few new variables that explain the most of the differences in observation, right? So we want to map the data from a high dimensional space to a lower dimensional space. And the importance is that we want to perform this task in a way that most of the information is preserved, all right? So I'm gonna cover 3 different dimension reduction methods. First one, which I think is the most widespread dimension reduction method, and the underlying method of many other like tasks that you might see in computational biology or data science and machine learning in general, is principal component analysis, or PCA. You might have heard it or seen some of the plots in different like manuscripts that you've read for biology. So again, like some of the clustering methods that you saw, for example, in single cell field or in many other omics areas. For example, when you're clustering your data, you don't perform the clustering on the raw samples. You first perform some sort of dimension reduction, and PCA is usually the most common one, and then perform the clustering within basically the PC space rather than the original space. Do you know why? Why would we do that? Like, what's the point of first transforming the data to a lower dimensional space and then clustering the data rather than just for applying clustering on the original data set directly?
Well, so about thousands of doesn't seem like very feasible.
Exactly. So the main issue is computational cost, right? So it becomes very slow if you want to apply it to the original data. Another issue that depends on the data set, but it might become important in data sets that are a little bit more noisy, is that first performing dimension reduction will essentially grab the core information within your data and get rid of the noise. So that's also a way to kind of like tackle maybe contamination or like some sort of noisy data set depending on the application. Yeah. All right. Okay, so let's go to the board again. Imagine we have 2 variables. Oh, this is a tough marker.
You like spray bottles.
That's okay.
I'm sorry.
Are there labs in the building? Is there one of those like ethanol spray bottles? I always would go steal one.
Oh, smart. I can see if any of the side ones have any
Yeah.
spray bottles.
All right. How many? Okay, here we have gene 1. We have gene 2, right? You have some dots. what are the dots here? Examples, right? Gene 1, gene 2, so it's a two-dimensional data set, right? Imagine I want to identify a line that captures the most, oh, thank you so much.
I'm gonna try and make a process. This is hand sanitizer.
Okay. Oh, thank you. Yeah, that looks well.
Okay, cool.
Good.
Okay, cool. I'll leave it
Yeah.
to you whenever you're ready. All right, there you go.
Yeah, great, thanks. Thank you. Yeah, so we want to draw a line. Again, we're going from higher dimensions to lower dimensions, right? So I want to identify one line, a new dimension, that captures the most information, highest amount of information in this data set. So I could draw it like this, like this, like this, or any ideas?
The line of best fit,
Is this a regression?
regression, yeah.
It's, yeah, it's very similar to regression. So, but it's not mathematically equivalent to regression. But yeah, exactly, your intuition is correct, right? So if I want to capture the highest amount of information here, I need to draw a line from right here to the middle. So this is essentially PC1, right? And what we're doing here intuitively is that we're trying to identify the axes that maximize the variance of the data, right? So what is variance? So it's like it has the highest spread of data on this axis. And if I want to, okay, so what would the value of each of these data points will be in my new dimension?
Maybe delta is off of it.
I essentially project each of these points to this new axis. And if this was gene 1, gene 2, and then sample 1, and whatever, sample n, what will it be converted to?
Table of deltas.
No, you don't usually kind of directly calculate the deltas here. That's again more of a regression type of problem. Again, I just want to, we're not gonna go through the mathematics of it, but the regression line will be very close to the PC, but it won't be overlaid on it, so they're not mathematically equivalent. And essentially what we will have here is PC1, and then again, sample 1 to sample n. And what did happen here? We went from 2 dimensions to 1 dimension, right? Dimensionality reduction. So that's the essence of PCA. And the underlying assumption of PCA is that variance is a best proxy for information. So axes that maximize the variance have the highest amount of information. And so this is a very simple 2-dimensional data set that we're dealing with. You either had kind of 2 genes. So in other examples, we usually have again like thousands of features, right? So one axis will not be enough usually. And in order to identify the next best axis to capture the highest amount of information, what PCA does is that, so PC2 will be orthogonal PC1, and so on, and PC3 will be orthogonal PC2, and you'll continue until needed. And we're gonna discuss how we usually identify the number.
Is there like a limited number of times that you could change orthogonally before you are back?
Yeah, so the dimension that you end up with, again, you usually stop way before then, but the maximum number of PCs that you identify is equivalent to the minimum of either of your dimensions of the matrix. So number of samples or number of features. And we'll see that in R as well. Okay, so this is just kind of the same thing we covered on the whiteboard. We have two variables. We want to kind of apply PCA to this data set, which, if you capture two PCs is not really particularly informative because, you haven't really performed dimension reduction. So kind of here you would kind of ignore PC2 and just look at PC1. But we're projecting the data into some new axes or dimensions. And just a terminology point here, each of these axes are called principal components, right? That's the PC that I was writing down over there. Okay, so the first principal component, or PC1, is the axis with maximum variance, right? And the more you go, the proportion of variance capture will be decreased. So the second PC, as you would expect, is the second highest variance captured in data, right? So we're gonna see some more examples. These lines that I've drawn here should be orthogonal to PCs. All right. Yeah, again, here in PCA, we're maximizing variance. And this is important because, so PCA is a member of a very larger family. It's called factorization method, matrix factorization methods. And depending on what method you're using, so it's a huge field in machine learning, different algorithms assume different metrics for capturing information, right? So we have ICA, NMF, and so on. So PCA is the one that particularly looks at variance. All right, so we call the process of projecting the data, which is mapping data to new set of dimensions, projection. And I'm gonna also introduce another terminology here, is that we have, when we perform PCA, yeah, let me go to the whiteboard. When we perform PCA, we end up with two matrices, right? So we have our data matrix. What didn't say is usually a tradition for representing columns, features, or variables, right? And samples are rows. Okay. So essentially, what PCA does here is that it splits this data into two lower dimensional matrices. Okay? So we have, I can make this dimension easier to distinguish from each other. We'll have features. Have samples or factors, and factors or features. Let's think of this as gene. I don't want to confuse you. Gene is very important. So our variables are genes. Okay? Which, when you're doing dimension evaluation, it does make sense, right? Because samples, these factors will go with each other and it will become sampled by genes again. Does that make sense? And I'm just introducing a terminology. So we call this matrix, it's samples by factors. We call it score matrix. And we call this matrix the loading matrix. So the loading matrix represents the relationship between factors and genes, and factors are our PCs here. And the score matrix represents the relationship between samples and. Does that make sense? So we have, until now, which of these two was I kind of which of these two, for example, in that plot over there, or this isn't a good example. Go to the next slide. So here, so which of these two matrices are being used here in this play example? We have dots, so this is the mesenchymal, and the other one, which was neural crest? That's neural crest, right? So mesenchymal and neural crest data sets, dots obviously represent samples. So which of these two matrices am I using? So we have PCs and samples. This score matrix, exactly. So we had two matrices, right? Samples and factors or PCs, and factors and genes, right? So if I'm visualizing PCs and samples, so my individual data points are sample, then I'm using this score matrix. Does that make sense for everyone? Is this clear? No? Okay, so we had this data. We applied some algorithm called the PCA. And it gave us two matrices, right? One of them is the dimension is factors and samples. So each of these values in this matrix essentially represents what is the score, what is the PC score, and this is, for example, PC1. What is the PC1 score in sample number 1, in sample number 2, in sample number 3, and so on.
And that's kind of like the distance to the line, like you're talking about, each one of those values on that.
Yeah, these are samples. Exactly.
The PC value will be like what you just drew on the line, like the distance to that line that you drew.
Yeah,
That's what
yeah.
the value
So
represents.
in the score matrix, because it's hard to clean up it. Oh, it's actually really helpful.
Feels very smart right now.
Yes.
She's like surprised. She's like, wow, it actually works. Well, ethanol got me thinking.
All right, so the score matrix was factors or scores, factor or PCs, sorry.
Okay.
And so that's the column here. And again, every matrix that we talked about can be transposed, right? So don't really think too much about column versus rows.
Yeah.
And here as rows, we have samples. So we have M1, M2, M3, NC1, NC2, NC3.
Right.
And PC1, PC2, PC3, and so on. It's a matrix,
The
right?
values in those columns are
Yeah, so
what?
3, 1, 5, and 0, like 6, 2.
But where are they coming from? Those values.
PCA data.
So, and that's like the distance to that PCA line, right? That's what those values are for each point, like we were talking about, like.
Not the, not the, it's the value of the projected. So we projected the data into this new axis, right? When I project this line here, it will have some value. But this is 0, 1, 2, 3, 4, and so on.
It's like a slope.
No, this is my new, this is my new coordinate system. I had an old coordinate system that had too many axes. I projected the data to a new space that has lower dimensions. And I have a totally new coordinate system, right? And the way that it works is that this, again, this line should have been orthogonal. You can map this here, right?
That distance, is that?
Not the distance, the value of it here.
Okay, on that line.
Yeah, on that line. For example, here, this would have some value, right? That's the new numbers that it. Okay?
Okay.
And I can visualize this, right? It's a new, let's call this 0 and PC1 PC2, and 3, 0, 1, 2, 3, 0. So this is my M1. M2 is PC1 and 6. This is M2, and so on. Right? So this will give me my new data, whatever the rest of things will be. I just showed you an actual example of it. We'll go back to that, right? So when I'm visualizing my data from PCA results, I'm using this score matrix, right? But then I might want to know, okay, what genes are captured in this PC1? Because this is some linear combination of my variables, right? I might, again, it has some applications in different bioinformatic tools, and we can then look at the loading matrix. Okay, so what's the dimensional loading matrix? What are the two parts? Here we have factors of PCs versus samples. Exactly. So that would be the loading. And each of these elements would represent what is the score of my gene in that PC, right? For example, PC1 might be really high in like albumin, some gene, right? Like this might have score 10, and some other gene, if we sort that list, like some other gene, I don't know, some mitochondrial gene might represent like 5, some other high score, right? And that could potentially help us interpret what each of these PCs are capturing. All right? Does that make sense to everyone? The main takeaway is that we have two matrices. And this can be, I went to a little bit more detail than like just looking at the output and applied version, because this is very important topic. You're gonna come across it many times in your, bioinformatics career. And it can be a little bit confusing to make sense of this. So just knowing a little bit of detail about how things work under the hood might become handy in the future. All right? Okay. Yeah. Yeah, so the maximum number would be the minimum of number of rows or columns of your data, depending on here. So if you have, I don't know, 100 samples and 10 variables, like 10 genes, it would be 10. The maximum number would be 10. Yeah.
What about when you want to mix them to
Like the number of PCs that you would want to use in yeah, we'll cover that. Yeah, there are methods for identifying the optimal number of PCs to use in different analysis. Yeah, we'll cover that both in the slides and the tutorial. All right, so it has many, again, different applications for both omics and non-omics data. Kind of tells us where the dominant structure in the data is. It's useful for identifying batches or unmeasured variable effects. So again, if you, for example, when you're visualizing the data using PC1 and PC2, and if you color the data points based on which batch they're coming from, and you see an obvious clustering pattern, it's kind of telling you that, okay, batches not well mixed in my data. All right, so that's very informative. You would want to figure out ways to get rid of that batch related variability. So again, that's one of the applications. And as we discussed earlier, it's very common in different machine learning methods to kind of reduce your feature set for more accurate modeling. Okay, so this is related to the noise reduction that we were discussing earlier. All right, so I'm quickly gonna cover the major functions, and then we'll discuss the more detailed part in the tutorial. Would you guys prefer if I cover the slides completely and then go back to the tutorial, or kind of like mix them together? All right, so the main function is that we use for PCA in R is PRComp. And here, as we kind of briefly discussed before, what is this T? Does anyone know? Transpose, right? So PRComp assumes that the columns are features and the rows are samples. So depending on how your matrix is defined, again, different methods have different assumptions. You can quickly just look at the help page. Just make sure that you're inputting the correct dimensions, because it will give you a result if you don't perform the transpose as well. So it's easy to make a mistake. And with STR, we can just look at the output. So we have standard deviation of each of the PCs. So this is again related to the variance explained. So which PC had the highest variance explained?
The first one.
The first one. You can see that there as well, right? So this is just like a toy example. You can see that the first PC is capturing the highest amount of variance. And then you have the rotation, which essentially is your loading values. It's hard to read here, but we'll look at it when we're at the tutorial. Center and scale essentially evaluate, like center is the mean of your PCs, or it's actually, I think it's the mean of your variables before applying PCA. And scale is just a logical value which tells you whether it applied scaling to the data before performing PC or not. Okay. And X is the score matrix that we were talking about. So again, which one was the score matrix? Factors versus what? Samples. Thank you. And what about the rotation, which was loading? The other one, right? So it would be genes. Loadings is for features versus factors. And in R, it calls it rotation.
Loading and there were two of them, right?
Score matrix.
Score matrix.
Yeah, score matrix is samples versus factors or pieces. Yeah. Right. Which rotation method? What rotation do you mean? Do you mean for improving interpretability? No, not by default. So I don't know if you're referring to the idea of rotations in factor analysis or not, but yeah, so that's actually something I was very much focused on in my graduate study. So like there are tons of different, we will not be covering that at all, but I understand that the terminology is confusing. There's a lot of overlap. And the reason for that is that historically these ideas were developed separately in different fields. So rotations mean tons of different things in, like, whether it was developed with statistics, like whether statisticians developed something, or computer scientists or mathematicians or physicists. So there's some history behind that. But rotations that I think you're referring to are the essentially, yeah, Varimax, Promax, Quartimax. They're essentially some extra step after performing PC that will help you improve interpretability. Meaning that the association between PCs and your variables will be more one-to-one. So you can kind of match PC1 with only a few genes, like that could be like, I don't know, markers of a specific cell type. So then you could say, okay, PC1 is capturing cell type identity of macrophages, for example. But we're not going to cover that today. But yeah, it's good to be aware of that those methods do exist. But they're post PCA, so it's kind of a separate idea. All right, we have standard deviation, loading values. we also have this summary function in R that kind of gives you some summary stats of whatever object that you're dealing with. So for PCA output object specifically gives you a table like this, when you have standard deviation, which is the same as what we saw in the previous slide, proportion of variance, and cumulative proportion. So proportion of variance is essentially how much, like, this value divided by sum of this whole first row, right? And cumulative is kind of like the first one is the same as itself. Then the second number is these two summed up together. The third one, these three, and so on. Cumulative, right? And at the end, it should sum up to 1, as you would expect. And as you can see, the amount of variance explained reduces as you go down with each component, right? Like 5.5, 1.7, and so on. You can look at the proportion if it's easier to read. You can also visualize this, right? So if I want to visualize this second row, what would my plot look like? Like what type of plot should I use? Yes, your head. I meant mostly like bar plot. And yes, the bar plot that visualizes that information is called script plot or elbow plot. And this goes back to your question, Mahshid, that you asked, how can we identify the optimal number of PCs to use in our analysis, right? So just to give you a little background, these parameters that we choose, they're called hyperparameters. And these are kind of open questions in general in machine learning field. Another hyperparameter that you might have came across today is like the number of clusters. So it's generally not always obvious that how many clusters should you ask your clustering algorithm to identify, right? So usually we use some intuition or prior knowledge based on the biology that we know to make decisions about this, or just try out different ones and see which one looks better. So these are some heuristic methods that can make us make more well-informed decisions about this hyperparameter choice. Okay, so again, the data is exactly this second row here. We're just representing them as bars, right? So the y-axis is percentage of explained variance, and the x-axis is PC number, like PC 1, 2, 3, 4, and so on, right? And as you kind of saw in the last slide, the percentage of explained variance kind of drops quickly, right? And depending on the data set that you're going to use, this slope could be more or less steep. So that's very much data set dependent. But definitely the pattern will look like this. So it will be a decreasing pattern by definition. And usually what people again, this is not a very rigid rule, but usually how people use this plot is that we look at the elbow. So the elbow here, it looks like it's PC2. So you can kind of think that the first two PCs will be good enough for me to perform my analysis on. So if it kind of looked like, I don't know, like there were higher, imagine the second and third and fourth all had high variances, and then it dropped suddenly on 5, then you would use the first 4 PCs, for example, right? Usually doesn't make a huge difference. Like after some point, like if you're choosing 20 or 22 PCs, again, like your visualization might change a little bit if you're running another, for example, algorithm on the PCs, but doesn't make too much of a difference. So no need to worry too much about that. All right, so how do we visualize the PCA results? I kind of visualize it already on the whiteboard, but this is just a tidier version. So this is the mouse data set again. We have 6 different, okay, so what are the dots here?
My sample.
Exactly, so dots are samples. And each of the, and what is the visualization is telling us, like the coloring and so on. Yeah, two different groups, and they're kind of like grouping with each other, right? So what does this tell you about PC1?
And there's lots of variance between the two groups.
Yeah, there are a lot of variability between the two groups. And what do you think? And like, if you look at the genes, how can I look at the genes that are captured with PC1? So I want to know, this PC1 is kind of like combination of set of genes, right? So how can I parse out that information from my PCA results? I covered it on the whiteboard. We have two matrices, right? Which matrix was used for this visualization? Score matrix. So if I want to figure out what genes are being captured in PC1, which matrix should I be using?
Yeah.
The other one, right? There aren't too many options. Loading matrix, right?
I just can't remember, I can't remember what the right. The one on the right.
I think my handwriting was also kind of
There's no
small.
to scale. and-
So yeah, this is
Right, let's see it. Thank you.
loading.
That helps.
Yeah, no worries. And this is score. This, gene.
Oh, so you can kind of-
Yeah. All right, how would I interpret PC1 again?
What is it trying to tell us?
Yeah, so that was a very valid hypothesis, right? So it's, you suggested that PC1 is capturing the variability between neural crest and mesenchymal groups. So then I went ask, okay, what are the genes that are capturing that variability?
We don't know, because it doesn't provide us with the gene.
It does though, you have two matrices.
Loading?
Yeah, you can look at the loading values, right? So you can look at PC1, you have a score for each of the genes in the, for PC1 and all the other PCs in the loading matrix. You can just sort those values and then look at, okay, what are the top ones? And there seem to be the positive side, seem to be capturing the neural crest, right? And that essentially tells you what are the neural crest associated genes. Again, this is exploratory analysis. I don't know if you guys have covered differential expression or not, no, right? So there are more like statistically rigorous ways to actually perform that analysis, but these type of, this unsupervised version is, they're kind of complementary. So this can give you some more like deeper insights about the data set that you're trying to analyze, right? So these are optional like analysis that you could potentially do if you wanted to dig deeper after visualization.
The table in the loading matrix looks like the score matrix is not like,
It's PCs versus genes.
just that, it looks like that. like
Yeah,
table
it's a
matrix.
matrix. It's a matrix. Yeah. We'll see that when we're in R. All right. So we're gonna quickly cover this alternative way of visualizing the data. But before that, just a quick summary of what we learned. So we have original data, we perform PCA, we have the loading matrix, and the weights show association between the original variables and PCs, and the score matrix, which was samples versus PCs, right? So this is the iris data set, if I remember correctly. So it's kind of give you information about different physical features of some plant families, right? So can someone tell me here, and again, this is here, I'm just like copy pasting the first 6 rows of that data set, but can someone tell me what are the samples and variables in this data set, in this original data?
Variables are.
Yeah, so like petal lengths, petal widths, and so on, right? Just plant like anatomy or whatever. And pardon?
It's like plant anatomy instead of gene.
Exactly. And the rows are just like different samples or different plants, right? That this scientist has gathered. So here, based on, you know, the legend and so on, what are each of these dots representing? Sample. So again, we do have this loading information, and you might want to know, okay, how can I add this information about my features, the relationship between my features and my PCs on top of my PCA plot, right? So that's essentially how people present this information. So here, each vectors are just the loading matrix values that we have, right? So for each of these features, we have 2 numbers, right? Which would be this dot, and it puts the center, it puts kind of like the center of the coordinate system for the vectors on the center of the PC plot, and it can be represented, and each of the features can be represented as a vector. And it kind of gives you a quick summary of the relationship between PCs and your features, which was captured in the loading, right? And also it can be kind of helpful for evaluating the relationship between features themselves. What does that mean is that, for example, in this example, what do you notice here by comparing the vector of petal lengths and petal widths? Yeah, they're very similar, right? It kind of tells me that these 2 features that I'm gathering are not super discriminated. Like they're very similar. They're relatively redundant, right? So if I was concerned about removing features, if I wanted to kind of not include all the features that I've gathered in my analysis, I could just choose one of them, right? But it seems like the sepal width is very different from the other ones, right? And it's relatively negatively correlated because it's on the other direction.
I just want to know that, so these vectors, is there a relationship between them and the clusters?
Yeah.
Steeple width is off towards that red cluster over there. Is there a relationship between those 2?
Yeah. So you can say that like this blue points, whatever, it's hard to read the plant names. No. I mean the dots. Virginica, right. It seems to be, they seem to be have higher values in petal lengths and petal widths, potentially. Yeah. Yeah.
And so then the bosa, that is, it has more likely bigger steeple width because it's worth.
Yeah.
But wouldn't you have like less sepal width because the sepal width is not, yeah, it's like on low and then it's not exactly on the dots.
Sorry, where?
Like petal length is very close to the blue dots. That means Virginica has high petal length.
The thing is, the distances are relative to the other dots. And as far as I'm aware, this is usually not very commonly interpreted. So usually you don't interpret the relationship between dots and vectors. You usually do the analysis kind of separately. So
Okay.
you evaluate the relationship between dots and PCs and then vectors and PCs. But it's kind of like providing all that information at once.
Yeah.
Yeah, because it can get a little bit confusing, especially with the gene expression information, because in omics, you essentially have so many features that these biplots become a little bit unreadable. For example, this is the example for what is it called? The mouse data set. And so what are each of these vectors? They're different genes, right? And the dots are mice samples. So it usually in your analysis, if you do that, it's just not very readable, just because how many features you have. But it is something that kind of like it's a common practice in PCA in general.
So this is like just another, this kind of matches the heat maps that we saw where the genes seem to be in two.
Yeah, that's an interesting observation. Yeah.
So you can like.
There-
cross-reference them.
Yeah. So there are two groups of genes that have relatively distinct expression patterns, right?
That's all you can really tell, like the positive or the negative.
Yeah, the positive and negative would kind of tell you that. So I don't remember if these were it. Yeah, so it kind of tells you that, okay, so these genes are more associated with neural crest, and these genes are more associated with mesodermal samples, or like these are mesodermal genes. What I meant by the interpretation is that it's the lengths of these are meaningful with respect to the dimensions. So the higher the length means that the gene has higher loading values for that PC. But interpreting that, okay, this is closer, like, is this closer to this or this gene? You know, the interpretation becomes like very loose.
Yeah.
It's not, it's over interpreting the plot. All right. Okay, so we just have very few slides left, and then we'll have a break and then the tutorial. All right, so we covered PCA, which was the toughest one. And again, I would argue that it's the most important dimension reduction that you need to know about. The limitation of PCA, though, is that it's a linear method. So if the pattern of separation between the samples is nonlinear, PCA will not be able to kind of identify axes to separate them from each other. So the mice data set that we saw was a simple data set. So the pattern was kind of like easily identifiable, but there are some more difficult and more complicated and nonlinear like gene expression patterns that you might come across. For example, in single cell transcriptomics, and I will show you some examples of that. If you just run PCA on your data and visualize it, it's very difficult to discriminate between cell types from each other, right? So people have developed more complicated and nonlinear methods. And one of the common ones is called TSNE, which I think was developed at U of T. And it stands for T stochastic neighbor embedding. And essentially, it's a nonlinear method and is developed for data sets that cannot be separated using a straight line. And what it tries to do is that tries to find fewer like dimensions that represent many variables while preserving the neighborhood distances. So you have the data set in your higher dimensions, and you can look into, okay, this data point number 1 is close to data point number 10, 11, 16, and 20. Okay, so I can measure these distances, and when I'm mapping that data into lower dimensions, I can try to preserve that distance. So I want the neighborhood of the individual data points to be similar between higher dimension and lower dimension. Does that make sense? So it has kind of a different mentality about preserving information from like what we saw with PCA. So it's very common and for visualization, again, especially in single-cell RNA-seq. Another one kind of issue with TC and also UMAP, which we'll cover next, is that these are stochastic methods. So PCA is solved analytically, meaning that it has one unique solution, right? But TSNE and UMAP and many other nonlinear methods, they're essentially solved numerically, meaning that they solve this very difficult nonlinear optimization problem iteratively. And when you rerun the method, you might get slightly different results. And to make your scripts more reproducible, it's a common best practice to set your seed. Because seed is essentially what is used for random number generation in many different different algorithms. And when you set that seed, you'll kind of like fix the random number generation and also fix the result of TSNE or UMAP or whatever stochastic method that you're using in your analysis method. So TSNE tends to focus more on local signals and local neighborhoods versus more global signals. And it's also a little bit, this is one of the major differences with UMAP that we'll also cover next. So this is what the TSNE result could look like on some two examples. So what are the dots here, do you think? So here we have TSNE1 and TSNE2, and what could dots represent here?
Sample.
Different samples. And what do you think these colors could potentially represent?
It looks like every sample has its own color.
Yes, the colors are like each sample is only colored once. It's not like divided, points are not divided based on colors. But this could potentially be a result of clustering, right? So you could potentially have a separate script for clustering your data and then visualize TSNE results using the clustering information that you have, right? And this is a single cell example that I was referring to. So on the right, we have TSNE result, and on the left, we have PCA results. And each dot, what is each dot representing here? And what are samples in single cell transcriptomics?
A cell.
A cell, an individual cell. So each dot is an individual cell. And what do you think they're colored based on? No, that would be sad if they were sampled. It would represent a very high batch effect. No, each dot is a cell, so it can't be gene, right?
It's like they're using types of receptors.
It's essentially different cell types, right? So one of them could be inhibitory neurons, the other one could be excitatory neurons, or like neurons from different regions of the brain and so on. Does that make sense? This is very common in single cell transcriptomics data representation. Okay. Okay, so the takeaway here, by the way, was that what do you notice when you're comparing PCA and TSNE? What we would ideally want to look at is, because then you could also visualize the expression of different genes over TSNE, right? So color them based on how much a gene is expressed. You would ideally like to have different groups for different cell types. Do we get that in PCA? No. So everything's kind of like mixed together, right? So it does not have the sufficient power to separate different cell types from each other, right? Whereas t-SNE is able to do that. That's kind of like the takeaway. So nonlinear methods could be helpful in that. All right, so one thing that we're gonna look into in R, so again, the function that we'll use, it's, not surprisingly called t-SNE. And what is this hyperparameter here? Perplexity, right? So essentially perplexity represents the neighborhood. Do you remember that I told you that what t-SNE essentially is doing is that in higher dimension, it looks at each individual point, look at its neighbors, right? So one hyperparameter here could be how many of its neighbors should it look into, right? So perplexity is essentially it's a proxy for the number of neighboring data points that it's looking into and try to preserve that in lower dimensions. Yeah, so determines how to balance attention to neighborhoods versus global structure. So the higher perplexity essentially means that we're looking into more global structure, right? Because we're looking at a lot of different cells close by. And this is how it would look like on our data set, which we have the tutorial for. So what do you notice here? More spread it out, right? So for lower perplexity, there are more dense clusters, and higher Vigo is more spread it out, which does intuitively make sense, right? Because it's kind of in lower perplexity, it's just looking into very few different neighbors and kind of trying to preserve that. So it's kind of make the clusters more tight. Again, we'll write the code for that. It's a hyperparameter. It's very much dependent on the example and the data set that you're dealing with. I can tell you that for single cell, for example, something around 30 is very common. But this is something that you would have to play with and identify like the best number based on what you know about the biology there. Yeah, it could be. Yeah. Because like obviously different single cell transcriptomics data sets are more similar to each other. Whereas again, if you're thinking about Netflix data set, right, it's like the data is fully different. All right, so the last method that we'll cover is UMAP, which stands for Uniform Manifold Approximation and Projection. Again, it's similar, very similar to t-SNE, and tries to conserve neighborhood similarity. It's better than at preserving global distances compared to t-SNE. So in t-SNE, it's not recommended to kind of try to interpret global distances from each other. For example, if one cluster is here, one cluster is here, and the other one is here, you can't really interpret that these two clusters are necessarily more similar to each other. You should kind of like interpret the results in a more local manner. But UMAP, the math behind it is kind of slightly different from t-SNE, and it's essentially better suited for capturing global variability compared to the t-SNE method. And this is the plot for UMAP, which we'll cover. Okay, that's it. These are the slides. And we're gonna take a break and be back for the tutorial.
Yep. All right, so why don't we take our half-hour break now?
Yeah.